We know now that the virus does its work by incorporating its DNA into that of the cell it infects. The purpose of this is to make the cell produce more virus, but in the process there is a side effect. Some virus DNA contains genes called oncogenes that cause cancer in the cells they infect. Research on this finally led to the discovery that normal human DNA contains oncogenes that are normally repressed, and that chromosomal abnormalities or mutations produced when the cells divide can activate these oncogenes, subsequently causing cancer. Not all humans' cancers are caused by viruses, but the evidence is becoming quite clear that all cancers are probably the result of this type of mutation in the DNA apparatus. The mechanism that causes cancer is a piece of DNA. This is, without a doubt, a triumph of technological medicine. But has it brought us any closer to a real cure for this ravaging disease? Unfortunately, at this time, the answer is no. The reason for this lies in the mechanistic view that living cell has a very limited ability to change or heal itself. Molecular biologists seem to view the cell as simply a container for DNA. We have already discussed the ability of cells to defy previous dogma by returning to their embryonic state in regeneration. It now appears that this process has a lot to do with cancer. Unfortunately, the promising avenue of exploration has not been followed. For this story, we must return again to the late 1800s. Regeneration in a cancer. Since cancer is characterized by unlimited growth of cells, one must ask whether this is solely the result of the oncogene activity or whether the oncogenes produce some more general change in the entire cell. One of the prime characteristics of cancer cells is that they are partially dedifferentiated, with the usual cancer cell being somewhat more like an embryonic cell than an adult one. However, this cancer cell is not as dedifferentiated as the cells in the regenerating blastema. It appears possible that this aspect of the cancer cell is what enables it to grow in an unlimited fashion and escape control of the normal body systems provided with an appropriate environment and continuous supply of food. Cancer cells are truly important. While this is not necessarily a desirable thing, I view it as a miracle. Under the circumstance, life can evade death. Certainly, there is something to be learned from this ability and from the relationship between cancer and differentiation. In 1877, Julius Frederick Cohenheim, one of the leading German pathologists of the time, suggested some interesting ideas on cancer. He was the first to observe that the cells of cancer were less mature than the cells from which they were derived. To him, they appeared to be almost but not quite like the cells in the embryo. Cohenheim proposed that cancers were caused by groups of embryonic cells, which he called embryonic rests, that were left over in the adult body. To find out why these resulted in cancerous growth, he made a startling conceptual leap. He proposed that there were control systems in living things that regulated cell growth, and that the control system in the adult was different from that in the embryo. The result was that the embryonic cells left over in the adult would not be regulated by the adult control system and would then grow in the uncontrolled fashion characteristic of cancer. More importantly, Cohenheim postulated that if the embryonic control system could be produced in a cancer, the cancerous cells would revert back to the totally embryonic state and then return by the normal process of dedifferentiation as normal cells. Cohenheim's embryonic rest theory is, of course, wrong, and his idea that the cancer cell could revert back to normal cell is very much at odds with present knowledge. Molecular biologists look at the DNA code in the cell as something fixed and immutable, except when an outside agent, such as a virus or carcinogen, interacts with the DNA itself. Therefore, anything that could have its effect upon the cell itself could not alter the DNA. To these scientists, then, the idea of cancer cells becoming normal is ridiculous. The only way to control cancer is to kill it, or to find a way to actually enter its DNA and alter it. Fortunately, long before we knew all this, a few brave souls looked at Cohenheim's crazy ideas In the 1920s and 1930s, a few experiments showed that chick embryo cells implanted into adult chickens became cancerous. However, these tests were so far outside the mainstream of scientific thought that no one paid much attention to them. In 1948, a biologist friend of mine, Dr. S. Merrill Rose, conceived a way to test Cohenheim's thesis. Rose postulated that when the salamander regenerated a limb, the original embryonic control systems were locally reactivated, producing dedifferentiation of the mature cells in the wound. The resultant regenerative growth was identical to a replay of the original embryonic growth of the same limb. The possibility occurred to Rose that if cancer cells were implanted into a regenerating salamander's limb, they could become normal. If Cohenheim was right. Since salamanders never normally develop cancer, Rose's first experiment was to transplant frog kidney tumor cells not known to be caused by a virus into the foreleg of some salamanders to make sure that they would take and act like a typical cancer. He found that the cells did become incorporated into the salamander, growing locally for a while as a visible tumor and then spreading in typical cancer fashion to the remainder of the body, ultimately killing the salamander. After Rose had proved that frog kidney tumor cells were cancerous to salamanders, he repeated the experiment. But this time, when the cells grew to the local tumor, he amputated the salamander's foreleg through the tumor, leaving cancer cells behind in the amputation stump. As the salamander began to regenerate the missing portion of its foreleg, the cancerous tumor's mass disappeared, merging with the developing blastema. When the forelegs were regenerated, they were normal and the tumor was gone. There was no evidence of cancer, and the animal lived normally thereafter. When the regenerated portion of the forelegs were examined microscopically, Rose could see nuclei of the original frog cancer cells and normal salamander cells of bone, muscle, and so on in the regenerate. He concluded that in the presence of a regenerative growth process, cancer cells could revert back to normal cells and become part of the growing blastema. In other words, Cohenheim had been right. Cancer cells were not locked irretrievably into malignant state. In the presence of embryonic control systems, they could return to normal. Unfortunately, this work was still outside the mainstream. Only a few scientists had duplicated this experiment since then. While they have all reported the same results that Rose did, no one has paid any attention. Clearly, something in the process of regeneration has the ability to speak to the cancer cell and cause it to rearrange its genetic apparatus so as to deactivate the oncogenes. Possibly the signal to dedifferentiate is stronger than the oncogene signal to reproduce. The fact that DC electrical currents play a significant role in regeneration, and also that cancers have extremely high DC electrical potentials, would seem to indicate that electrical factors may be involved. Some attempts have been made to study this idea, and in later chapters, we will discuss the relationship between electricity and cancer. Since cancer cells have become normal cells in this fashion, shouldn't we be studying the process as a biological way to control the disease rather than continuing to try to kill the cancer cells? In a guest editorial in the journal Growth, Dr. Alexander Wolski strongly suggested a return to basic biological research on the relationship between cancer and regeneration. He also spoke of the Vietnamization of the war on cancer, the expenditure of enormous amounts of money with no effect on the enormous losses of life. Clearly, it is time for a change in outlook. Cancer and the rest of the body. The discovery of the mechanism behind cancer seems to have done little to dispel the idea that cancer is somehow something foreign within the body and that the body has little to do with it. This notion, coupled with Ehrlich's idea of a magical bullet for each disease, has led medical science down the road of devising chemical agents to kill cancer cells. This is not an easy task. The cancer cells change, though they are still our cells. Not only are chemotherapeutic agents against cancer toxic to the rest of the body, producing depression of the immune system, but many of them are themselves carcinogens. Only in recent years has the idea that the body has defenses of its own agent against cancer become popular. The roots of this concept are also buried in the late 1800s. At that time, Dr. William Coley, a surgeon at New York's Memorial Hospital, noted another puzzling fact about cancer. Many of his patients with cancer became completely cured if they developed this very serious life-threatening infection and survived. Coley postulated that the infection had activated immune resistance mechanisms in the patient, and that after the infection had literally brought the patient to death's door, the activated immune system not only successfully fought off the infection, but destroyed the cancer as well. Coley mixed up a combination of the worst bacterial toxins he could find and began injecting them into patients with inoperable cancers. Some died from the treatment and some did not show any remission of the cancer, but many went on to a complete cure. In 1975, a review of 186 patients treated with Coley's toxin showed cures in 105 cases, a much better success rate than any other treatment then or now available. Despite these promising results, no large-scale clinical studies have ever been done, and Coley's idea has been followed up in only a few laboratories. This laboratory work, however, has centered on the mechanistic concept that either the bacteria or the doctor must have produced one specific chemical that killed the cancer cells selectively without harming the body's normal cells. This work has led to the discovery of a chemical in the bacteria's cell membrane that causes the body to produce another chemical, one that, when injected into mice with implanted cancer cell, results in death of the tumor cells alone. The tumors turn black and liquefy. The chemical involved is called tumor necrosis factor, TNF. At present, there is considerable interest in producing this chemical by means of recombinant DNA techniques so that it can be used in human patients with cancer. However, there are problems with this approach. First of all, Coley's patients experienced a disappearance of their tumors. They did not turn black and fall off. TNF thus appears to be something else entirely. It has been noted recently that TNF has been studied in other laboratories where it has been given another name, endotoxin or cachexin, 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 cachexin. In this work, endotoxin was associated with the extremely serious and often fatal clinical condition known as toxic shock syndrome. In this condition, profound shock and death occur within few hours of the onset of a bacterial infection. The chemical responsible for endotoxin, cachexin, the same chemical as TNF. So the chemical pathway that is now being followed has produced another single agent that appears to be even more toxic to the human body than the synthetic anti-cancer drugs now in use. Why this chemical produces acute toxic shock syndrome in one case, necrosis of tumor cells in another, is indicative only of the fact that other factors within the body are operating. Advocates of scientific medicine still seem to believe that once a cancer cell, always a cancer cell. So the only treatment alternatives possible are to kill the cell, preferable with a single chemical agent or to surgically remove it. The vital role played by the body's own resistance factor is still ignored in this approach. It seems to me to be the height of the folly to treat cancer with agents that decrease these innate resistant factors. Resistance to cancer by immune system is not the only mystery left unexplored by modern technological research. Later on, we will discuss such strange occurrences as spontaneous remissions in which, for no apparent reason, cancers disappear. And we will explore the profoundly important role played by psychological factors in cancer. At the very least, the ability of cancer cells to revert to normal and the innate resistance factors of the total body should be thoroughly investigated as possible biological cures for this highly feared disease. Such an approach would seem imperative in view of a recent thought-provoking article, Progress Against Cancer, by Dr. John C. Baylor, a respected medical statistician, and E.M. Smith. We are losing the war against cancer, Baylor writes. Basing this conclusion on a thorough statistical analysis of the years between 1950 and 1982. That analysis showed the incidence of cancer to have increased steadily despite improvements in diagnosis and treatment. The mysteries of growth seem to be yielding to the concepts of electronic biological control systems. We are now beginning to understand how almost vanishingly small electrical currents act upon certain cells to produce healing, and in some instances to cause cancer cells to return to normal. While this is of obvious importance of clinical medicine, providing us with the ability to actually manipulate the growth process at will, it is of even greater importance of the success of the control system theory, which can be applied to many other areas of life. This is not a return to mysterious vitalism, but rather a logical extension of the mechanistic concept that reintroduces the ancient idea of life energy in a scientifically valid fashion. The morphogenic field, blueprint for growth. After both the input signals of injury and output signals that stimulate healing were found to be parts of an electrical control system, we were left with a problem. How was the new growth told what kind of tissue to become? A simple cut on the finger is easy. All that's needed is to close it by fibrosis. But consider the salamander's regeneration of a limb. Not only is it necessary for something to tell the blastema what to become, but the veritable mountain of information characterizing that specific extremity has to be transmitted to it as well. As I noted before, the salamander's foreleg is the counterpart of a human arm, right down to the tiny bones in its fingers. All the information on what cells go into the organized tissues, how the tissues fit together on the bones, joints, nerves, ligaments, blood vessels, and so on, has to be transmitted to the site of injury. Since the instructions relate to new growth to the total body, the system providing the instructions has to be part of the total body. What is it and where is it? Is it a part of the brain? The 19th century embryologists recognized this problem. In an attempt to solve it, they devised a few simple experiments concerning the blastema. What they found revealed what an amazing and even more inexplicable complexity. It is a simple matter to remove the blastema and transplant it somewhere else on the salamander's body, where it will grow then. If the scientists removed the blastema from a foreleg amputation before the 10th day after amputation and transplanted it near the intact hind leg, it would grow into a hind leg. The same blastema transplanted next to the tail became a tail. The embryologists thus concluded that there were body inductive areas or morphogenic fields that contained the information that specified what was to grow in that area. These experiments revealed a very important fact. There is some mechanism within the living salamander that contains an overall plan for salamander's body and provides the information that instructs the blastema what tissue it should construct. That was a monumentally important piece of information, but other experiments made it more important. The embryologists next found that if they waited until after the 10th day to do a transplant, the foreleg blastema became a foreleg, no matter where it was transplanted. Somehow, in a very short period of time, the necessary instructions to make a terrible complex structure had been transmitted to the unimpressive little lump of primitive cells. To make matters even more complicated, it was found that the nervous system, only means then known for transmitting such information, was not involved in the normal fashion. While the local nerves would grow into the transplant before it began to grow itself, both these nerves and those going to the amputation stump were completely silent throughout this period of information transfer. That is, they transmitted no information by means of nerve impulses. It was obvious that chemical messengers were totally unable to convey this much organizational complexity. Since the nervous system was so excluded, there had to be something else, something similar to the morphogenic field that can contain within itself the entire organizational plan. Since DC electrical currents had been shown to be deeply involved in regeneration, could such field produced by such a current contain this information? The answer is a qualified yes. Even the simplest electrical field has a high degree of organizational complexity. If, for example, you place two electrodes in a petri dish filled with a mild salt solution and then connect them to a small battery, a current will flow between the electrodes. When you look at the pattern of the current itself, it's not simply a stream of charge carriers running directly between the two electrodes, but a spreading out field composed of changing values of current and voltage. A large amount of information may be contained in a field such as this. Each point in the field may be characterized by a unique set of values of voltage, current strength, and current direction. While each point would therefore have a unique set of coordinates, this in itself is not enough to contain the entire blueprint for a complex extremity. The field provides only positional information. However, the field must act upon the primitive cells of the blastema. Each of this contains a large amount of information. It's possible that the field serves only as a trigger to instruct the cells to follow certain routines in development. However, the electrical field concept of the morphogenic field would seem to be at least a good starting point from which to embark on a course of study. If local electrical currents and the voltages controlling healing are not simply local phenomena, but are instead part of a larger total body DC system serving as a morphogenic field, there must be a second nervous system, one that controls the most primitive functions and that probably predates the nervous system with which we are so familiar. The existence of such system would force us to change our ideas of how the brain works. The dual nervous system uncovered, analog and digital controls. It sounds like quite revolutionary and perhaps even rather fringy to say that there is a second hidden part to the brain. While we do know a great deal about how the brain and nervous system work, there are some major questions that have not been answered. I have always believed that our most important roadblock in this area is that we consider all brain functions to be based on one mechanism, the nerve impulse. This is a very sophisticated mechanism in its own right. When it is used to transmit information, it then becomes an extremely complicated mechanism. The best way to approach this problem is to compare currently accepted mechanism of information transfer in the nervous system to a digital computer. In both the computer and the brain, the basic signal is a digital one, a single pulse. The information is coded by a number of pulses per unit of time, where the pulses go, and whether or not there are more than one channel of pulses feeding into an area. Our senses smell, taste, hearing, sight, and touch work by means of this type of pulse. The digital computer on my desk works in the same way. The digital system works extremely fast and it can transmit large amounts of information as digital bits of data. I have difficulty believing that the first living organisms used this same type of system. It would seem that they must have had a much simpler mechanism for transmitting information. The first living organism did not have eyes, ears, or other such sensory organs, and it wasn't much concerned with thinking great thoughts. What it needed was a way to know when it was injured and the means to efficiently repair itself, along with the ability to relate to important aspects of its environment and other living organisms. In short, it must have been rather unimpressive and totally lacking in complexity, since there was no requirement for quick action or for transmission of large amounts of sophisticated information. It could make do a much simpler system of analog data transmission and control. Before digital computers, there were analog computers. These operated in exactly the way I believed first living organisms did. The analog computer works by means of simple DC currents, with the information coded by the strength of the current, its direction or flow, and slow wave-like variations in its strength. While this system is slow and is incapable of transmitting large amounts of data, it's extremely precise and works well for what it's designed to do. Today, some of the most sophisticated computers are actually hybrid computers containing both analog and digital components. If the first living organisms used an analog type of data transmission, and if such system is still used for injury repair, then the entire analog system must be present. Although unseen as a part of our entire data transmission and control system, the brain may thus operate as a hybrid computer and with very important analog part that operates by means of DC electrical currents. This concept provides us with the entirely new way to look at the old brain-mind-body conundrum. My first experiments involved searching for DC electrical potentials in the uninjured salamander. Similar searches had been conducted in the past, notably by Professor Harold S. Burr at Yale in the 1920s and 30s. Using the best measuring devices available, he found DC potentials organized in a simple front to back fashion, with the salamander's nose being negative and the tip of the tail positive. When I began my work some years later, I benefited from the technical advances in instrumentation that had been made during World War II. Consequently, I found a more complex pattern that appeared to be related to the total nervous system from the brain to the end of the furthest nerve fiber. The nerve cells of the salamander are concentrated in the three places: the brain, cervical enlargement, and the lumbar enlargement. All are connected by the spinal cord, and from enlargement, nerve fibers go out to the periphery. Some of the fibers from the brain go to the special sense organs, but most go down the spinal cord to the other two enlargements. Fibers from the cervical enlargement go to the forelegs, while those from the lumbar enlargement go to the hind legs, with a few to the tail. The arrangement of our own central nervous system is basically the same. We found that in the awake, unanesthetized salamander, the three collections of nerve cells were electrically positive, and the nerves from them were more and more negative as our measuring electrode passes further along the limbs, or the DC potential seemed to be produced by the nerve cells themselves, because cutting the nerves to a limb in the lightly anesthetized salamander reduced the DC potentials in that limb to zero. However, the level activity of the nerve cells seemed to be related to the state of the entire DC system. For example, general anesthesia resulted in drops in the DC levels, starting with the brain and then proceeding through the rest of the nervous system. Finally, in deep anesthesia, all potentials were zero and even slightly reversed. We then found that the direction of the flow of the DC current in the motor nerves, those that make muscles move, was most often opposite to that in the sensory nerves. However, in both cases, we were able to show that the electrical current was semiconducting and not ionic. The method we use demonstrates a unique relationship between semiconducting electrical currents and magnetic fields, one that will later be shown to be important. If there's electrical current flowing between point A and B, you will put a strong magnetic field at right angles to the line of current flow. There will be a pull on the charge carriers from the magnetic field. If the current is electrons flowing along the wire, nothing much will happen because the electrons are tightly oiled or constrained within the metal. If the current is ionic, again, nothing much will happen unless you use an enormously strong magnetic field, because the ions are large objects and move slowly. However, if the current flow is composed of electrons or holes in a semiconductor, the charge carriers move very easily, and many will pile up in the magnetic field. If you then put two electrodes on the conductor so that the piled up electrons can flow into another circuit, a respectable voltage called the Hall voltage can be measured in that current. I measured the respectable Hall voltages from salamander's forelegs, voltages that could have come from the semiconducting current carried by the nerves. So there really is semiconducting DC system operating, at least in the nerve fibers in the limbs. What of the brain? Looking back in the scientific literature, I found that in the 1930s, several eminent neurophysiologists had measured DC voltages in the brain and had shown that these actually controlled the level of activity of the nerve cells in the brain. While this work was of obvious importance to the understanding of how the brain worked, it was essentially lost as instruments for easily and accurately measuring for nerve impulses came into use and difficult techniques needed for measuring DC voltages fell into disuse. Besides, everyone knew that the nerve impulses was where the action was. So why waste time measuring something that didn't make any sense? I decided to reopen this controversy, but from the point of view of the total body DC field. Since general anesthesia produced a drop in the normal front to back negative DC field across the brain first and then through the rest of the nervous system, I postulated that the chemical anesthetic actually worked by causing this change in the DC system. Therefore, it should be possible to anesthetize a salamander simply by placing electrodes on the front and back of its head and passing a small current through the head in a back to front direction. In order to prove that the animal was anesthetized and not electrocuted, even though I was going to use a very small current, I decided to measure the salamander's brain waves before and during the time the current was on. It was very exciting to watch the electroencephalogram EEG change from typical awake pattern of 8 to 10 pulses per second to the slow waves of 2 to 3 per second that are characteristic of deep anesthesia when only a few microamperes of current were applied with the frontal electrode positive. Later, it was able to show that the currents were in the brain were like the currents in the peripheral nerves semiconducting. With no external source of reverse current applied to the head, a magnetic field of 3000 Gauss applied at right angles to the front to back line of normal current flow completely stopped the electrical current in the brain and produced the identical slow wave pattern in the EEG. A DC analog system of data transmission and control is, in fact, hidden within the central nervous system. It had seemed unlikely to neurophysiologists in the 1930s that nerve cells had within themselves two such completely different electrical mechanisms of action. They therefore postulated that it was the other cells of the brain, the perineural cells, that generated this DC electrical activity. We commonly think of the brain as being packed with billions of nerve cells. While there are billions of nerve cells in the brain, there are as many, if not more, perineural cells. In fact, these cells are found wherever nerve cells or nerve fibers are found. They come in a variety of types, the most common of which are glial cells in the brain and spinal cord and the Schwann cells encasing the peripheral nerves. In the brain, the nerve cells can be likened to raisins in a pudding of perineural cells. All the perineural cells are related and arise embryologically from the same basic cell line, the ectoderm, and the nerve cells and skin cells. Anatomically, the perineural cells appear to be a system of their own. The Schwann cells surround a fine terminal branch of nerve in the fingertip and is in contact via the other Schwann cells along the entire nerve. With the Schwann cells in the spinal cord, which in turn are contact with the glial cells of the brain, every nerve cell or nerve fiber is surrounded by perineural cells of one type or another. If there were a way to dissolve the nerve cells, you would still see the brain, spinal cord, and nerves apparently intact. The function of the perineural cells has been something of a mystery. Most theories have ranged from seeing these cells as packing material that protects nerve cells to seeing them as providers of nourishment of nerves. However, it has been recently shown that the perineural cells can generate electrical potentials and pass them along from one to another. The possibility has never been seriously considered that the perineural cell system, extending throughout the whole body exactly as nerves do, functions as a primitive communication system. Uncovering the hidden cellular communication system. The relationship between the perineural cells and the DC system was discovered accidentally after one of my students became interested in the non-unions of fractures. Strangely, this seemed to be a condition unique to human beings because it was practically impossible to produce the same thing in animals. This student thought that if he fractured the tibia of a rat at the same time he cut the nerve supply to the leg, a non-unit of the fracture might result. The first experiment was a failure. The fracture in the rats healed only a few days slower than normal. I suggested to the student that in the next experiment he cut the nerve supply 2 days before fracturing the tibia. This group of rats healed their fractures slightly faster than the first group, but still slower than normal. This was getting interesting, so we did a third experiment in which the nerve supply was cut 5 days before the fracture. The rats healed their fractures in normal time. Obviously, something had happened during the 5 days that had restored normal healing. It could not have been regrowth of the nerve to the leg because this would take at least 1 month, and the rats in the third experiment had no functional use of these limbs, even after the fractures had healed. Fortunately, the student was a good investigator, and he had saved the entire legs of the animals as experimental specimens. We went back and dissected out the nerve supply in the third experimental group. We found that while the nerve had just begun to grow, there was a strand of something connected across the gap in the nerve. Under the microscope, we found that the strand was composed of tubes of Schwann cells that would later contain the regrown individual nerve fibers. The perineural cells, in this case, the Schwann cells, carry the electrical signals that cause fractures to heal. The nerves has nothing to do with it. It seemed justified to postulate that the DC system we were studying resided in the perineural system. If this were so, the perineural cells were more primitive than the nerve cells and likely represented the more primitive analog data transmission and control system. However, this implied that the perineural cells were the precursors for the nerve cells, and this was totally against the mechanistic idea that cell lines were fixed and non-changeable. I decided to do a simple experiment. I grew perineural cells in culture alone with no nerve cells present. After a week or so, these cells had grown into interesting swirling patterns. When I looked carefully, I could see an occasional typical nerve cell mixed in the perineural cells. While this does not absolutely prove that the perineural cells can transform into nerve cells, it does tend to support this notion. Interest in the DC system, particularly in the interrelationships with nerve cells in the brain, has been increasing over the past few years. Recent experiments have shown that when a human subject is told to make a certain muscular movement after being given a signal, there is an increase in negative DC after the signal, but that this occurs almost a half a second before the muscular action is performed. It appears that the DC is somehow involved in getting the neurons ready to fire the command to move the muscles. This phenomenon, which has become known as the readiness potential, seems to imply that the DC system commands the nerve impulses system. In a recent review article, Dr. Benjamin Libet of the University of California, a longtime researcher in the DC potential area, presented even more startling data. He was concerned with which came first, the readiness potential shift or the decision itself. Libet's experiments indicated that the readiness potential preceded the decision. In his words, the brain seemed to have a mind of its own. However, one wishes to interpret this. It seems clear that the DC potential system in the brain is activated prior to the nerve impulse system, and that the later may depend upon the former being in the particular electronic state. The DC system thus appears to be, in fact, the place where the actual command decision is made. If we combine all of the above observations, we can only conclude that more primitive analog data transmission and control systems still exist in the body, located in the perineural cells and transmitting information by means of the flow of the semiconducting DC electrical current. The system appears to have been the original data transmission and control system present in the earliest living organisms. It senses injury and controls repair, and it may serve as the morphogenic field itself. It controls the activity of body cells by producing DC electrical environments in their vicinity. It also appears to be the primary system in the brain, controlling actions of neurons in their generation and receipt of nerve impulses. In this fashion, it regulates our level of consciousness and appears to be related to decision-making processes. The startling conclusion leads to more questions. Is analog DC system smarter than the nerve impulse system? Does it regulate and control the digital nerve impulse system in the same fashion that we control and use our personal computers? Is it the seat of thought, logic, and memory? Can we control its operations and heal ourselves? These are intriguing questions, but before we can answer any of them, we have to add some unexpected and important complexities to this dual brain system, the magnetic connection.